on the main stage, we will be joined by our next speaker, who is a research scholar at the Future of Humanity Institute and a medical doctor. And today, she will provide an introduction to global catastrophic biological risks and make the case for biosecurity being an important EA cause area. So please welcome up on stage Dr. Cassidy Nelson.
Great, thank you. So yes, today I'm going to speak with you about biosecurity and the research field of global catastrophic biological risks. Thanks for coming to my talk. Just to begin, though, I do think it's very difficult, especially in the modern era and in the Western world, especially to think about infectious diseases in the way that this research field thinks about them. There's been a history of us being very much affected by infectious diseases, but it's not something that you see every day in the Western world. We now are much more likely to die of non-communicable diseases, such as ischemic heart disease and stroke, than we are to die of communicable diseases. However, that wasn't always the case. And this is Janet Parker, who in 1978 became the world's last victim of smallpox. Smallpox in the 20th century alone, from 1900 to 1978, ending with Janet's death, killed more people than all wars in the 20th century combined. 300 to 500 million people versus 187 million. And I think I get quite concerned about this because I worry that things such as smallpox and other new emerging diseases are something that we might have to face this century. So in my talk, just to give you an overview, the goals I'm going to have are just to give you an overview of biosecurity. I want to make a case for it being an important EA cause area. I want to leave you with some key messages that you can take home. And if you're interested in this area, I want to give you some ways that you could have an impact. Give you some suggestions about how to do so. So just to begin with, there's a lot of different kind of preconceptions about the word biosecurity, so I think it's good to just lay it out as a definition. Biosecurity encompasses the measures and procedures we use to protect populations from biological substances, and that includes infectious diseases. GCBRs have been defined in 2017 by the Johns Hopkins Center for Health Security with this definition, which I think is quite good, but it is quite cumbersome. So just as a kind of more succinct summary, GCBRs are those events caused by biological agents that could negatively and permanently alter the long-term trajectory of humanity or result in human extinction. And now, I don't have time in my talk today to make the case for long-termism, but I do think it's really important. If you're interested in this, and I hope you are, Aaron Ballender is doing a talk later today making the case for long-termism. But just in short, the reason that I care about GCBRs and existential risks is because events that occur that affect people today are by no means not tragic. However, the difference with GCBRs or X-risks, especially by this definition, is that it affects future people as well too. So if you have an event that's going to alter the long-term trajectory of our species or end our species through extinction, you're not just affecting the people that you affect today, but you're affecting people in hundreds, thousands, millions of years from now. You could end up affecting trillions of potential human lives. And that's why I think this area and reducing risks in this area is something that should be a priority. And GCBRs are just one subset of catastrophic risks. This is a book by Nick Bostrom. And it lays out other risks as well too, so by no means GCBRs are the only risks that face our species. But what makes them unique is that biological agents are not limited to just where they initially originate. So diseases have this unique property of being able to spread human to human. What that means is that if you have a single point source, you can actually have widespread geographical dispersion among populations in today's age very, very quite quickly with air travel, whereas other types of risks are more confined and a little bit harder to actually spread depending on the type. It also requires a functioning medical and public health system as well too, because biological agents incapacitate their hosts. And so I do think biological agents, for a few reasons, are quite a scary thing to start considering in terms of catastrophic risks. So what are some examples of GCBRs? There thankfully hasn't been any examples of GCBRs in our history, at least by the definition that I gave you before. There arguably have been some near GCBR events, such as the Plague of Justinian or the Black Death, or 100 years ago, the Spanish flu, which killed 50 to 100 million people. And these undoubtedly caused widespread suffering at the times that they occurred. However, they didn't threaten our long-term trajectory as a species, and they didn't threaten human extinction. And for that reason, they didn't meet the definition of a GCBR. 100 years ago, 50 to 100 million people died of Spanish flu, and that was more than World War I, which was going on at the same time. And it's unclear to me today whether if we were faced with a similar situation, if we'd fare much better. On the one hand, we have things such as antibiotics and supportive care. Antibiotics are very important because most people actually died of secondary bacterial infections during Spanish influenza, not of the viral infection itself. But at the same time, can our health systems cope with that number of patients? It's not clear to me around the world if it's possible to rise up to the levels that we'd need to. And this changes as well too when we think about not things that we understand quite well, like influenza, but new emerging infections, which we might not have much information about at all when they first begin as an outbreak. And there's some, such as Bill Gates, who in 2017 wrote this article in Time magazine, making the case that we're not ready for the next pandemic. This is talking about natural pandemics for the most part, but it's something that a lot of health security officials around the world also agree with, that current global health security is inadequate to deal with the next infectious disease outbreak that affects the global community. And it's not because money's not being spent in this area. So biosecurity does attract a large budget. This is figures from the US Federal Health Security funding of 2018. And you can see that the biosecurity allocation is $1.5 billion US dollars. Pandemics and emerging infectious diseases, more than $1.3 billion. However, I'd make an argument that most of this money is being spent on things that affect the left-hand side of this graph. So things that will affect tens to hundreds to thousands of people, but very few that could affect what we care about here of global catastrophic risks or existential risks. And it's unclear to me that money spent on preventing small-scale events immediately scales up to preventing larger-scale events. And I do want to make it clear that as you go from left to right on this graph, the likelihood of such events occurring decreases very quickly. The idea of an existential risk or a GCR is much less likely to occur in any given year in this century than smaller scale events. However, I'd argue, based on the impact that these events can have, that they are worthy of our consideration and that we should consider larger scale, lower probability events because they are something that we should be working to prevent. And my colleagues at FHI agree with this. They released, two of my colleagues released this report on biosecurity cost effectiveness back in 2017. And in it, what they did is compare cost spending. If you decide to spend money on reducing a event that could kill thousands or tens to thousands of people versus if you end up spending it on an existential risk event. And so they had three models for the existential risk events with different estimates. And they actually compared not the exact same risk reduction. As you can see, they've compared for the smaller events, a 20% risk reduction versus only a tiny 1% risk reduction for the existential risk. But because of the number of lives that are at stake, if you decide that in the same way that we don't want to discriminate based on where people are in space, so where you are geographically, if you decide that we don't want to discriminate where people are in time, you realize that if caring about future lives and incorporating them into your equations, you do actually get very cost-effective bang for your buck if you start spending money on trying to prevent risks that could have threatened our future. So I've been talking pretty abstractly about biological risks. The Johns Hopkins Center for Health Security last year, 2018, released this report where they tried to characterize what would a pandemic pathogen look like if it was to emerge. And what they found is that viruses are much more likely to result in a pandemic than bacteria or fungi. They also found RNA viruses are much more of a risk than DNA. That's due to their mutation rate. And also that the respiratory route of spread was the most likely to have a pandemic risk associated with it. All the same, natural pathogens are quite limited by various different constraints, mostly evolutionary derived constraints. Things such as optimal virulence. And what this graph shows is common pathogens looking at the case fatality rate over the basic reproductive number, R naught. And R naught is a measure of the average number of infections each case will generate. And if you look at that, they're inversely correlated. As you increase the transmissibility, so the number of infections that will result from a single case, it seems that the lethality of the pathogen does go down, and this seems to hold true across a variety of different pathogens. However, nature's limitations are not something that we necessarily can hold to anymore, and that's something that makes me quite concerned going forward. So this is a common saying among a lot of health security or health officials that because of the ability of evolution to generate deadly pathogens, nature itself is the worst bioterrorist. I don't think this is the case for our century going forward, and I want to make some arguments why. So there's three types, generally speaking, of GCBR scenarios that we care about. We look at natural pandemics, accidental release scenarios, this is from laboratories, and deliberate release scenarios, deliberate attacks. And I would make the argument that as you go down this list, the probability of this occurring in any given year does decrease. I'm not saying that deliberate attacks are anywhere near as likely as a naturally occurring pandemic. However, I would argue that the impact of such events potentially could be a lot worse than what nature is able to produce. And I think this becomes the case even more so if you start thinking about engineered pathogens. And so engineered pathogens meaning human modified pathogens that make them either more transmissible or more deadly or a combination of the two. I think this is even more so the case in that last category there. So people that intentionally do this with the intention of killing a lot of humans. So this is a quote from Dr. Eckhard Wimmer in 2002, him and his group in New York synthesized the first live infectious virus, poliovirus. And he said, after he published his research, that you no longer need the real thing in order to make the virus and propagate it. He said in some interviews following the research release that one of his justifications for doing it was that he wanted to show that bioterrorists did not need access to natural pathogen in order to make bioweapons. And it's true. It's true nowadays that you do not need access, unlike in the past, to natural pathogens to make new pathogens, because you are able, from genetic code, to be able to synthesize your own. And Dr. Edward Wimmer was the first with poliovirus, which is a small virus, 7,500 nucleotides long. But he was able to show that he was able to create and it was able to be infective to cells. So in the last 20 years, there's been a big debate raged about research that can be done which has both good uses for science as well as could be used for harm. And this is usually bundled up in the term dual-use research or dual-use research of concern. And in 2007, the US National Sciences Advisory Board for Biosecurity categorized seven research areas that they were particularly concerned about when it came to working with pathogens in research and things that could be done to modify them. And they were worried about this, about people not only doing work in this area, but publishing in this area as well too. And it turns out that there's published examples of all of these different ways that you can modify pathogens. And I'm just going to talk through a few now. So in 2012, there were two separate labs that worked with avian influenza. So this is an influenza A H5N1 virus. What they did was use ferret models, which is a common model to use in influenza because they have similar respiratory systems to humans. Their cells get infected with influenza the same way ours do. And avian influenza is a scary pathogen in that it's quite deadly to humans. However, it very rarely passes human to human, so it's very difficult for it to actually have sustained transmission between mammals. Most people who get avian influenza work either in intensive agriculture with different types of birds, or they work with wild bird populations. But very few cases have ever transferred onwards to another human. What this group showed, though, was that using serial passaging of the virus back and forth between ferrets, they were able to get the virus to mutate. And with as few as 5 amino acid changes, they were able to get it to be airborne transmissible between ferrets. So first case of avian influenza that was shown to be airborne transmissible between mammals. And this caused a lot of controversy at the time, and there was a lot of debate about whether or not this type of research should be released. On the one hand, if it's that easy for a virus to evolve to become airborne transmissible, should the scientific community know about it? On the other, should you publish the methodology of how you go about doing this, and then say which amino acids you need to change to be able to do this? There's been a lot of disagreement between scientific communities as well as biosecurity communities about the risks that such research poses to our global community. And it seems that there's been a massive change in the landscape of what we're able to do in general with research. So the cost of gene synthesis compared with even just 20, 30 years ago is just massively decreased. This is a logarithmic scale on the y-axis there. So you have the capability of synthesizing genes in a way that you just financially had a lot of restrictions before. This is for research labs. This is for people who are interested in exploring various different ways about both learning about pathogens as well as about synthesizing new organisms. And so we have had this kind of changing playfield where there's more actors that are able to enter this space. And one example of this is the horsepox virus synthesis that there was a publication last year of 2 Canadian researchers that were able to synthesize an eradicated pox virus. And so pox family of viruses are really well known for being very difficult to even think about synthesizing. They're very large. Like I said before, there's 7,500 nucleotides in the poliovirus, the first virus ever synthesized. These ones have more than 150,000 base pairs. And horsepox doesn't infect humans. Horsepox is not a danger to us. But it has a closely related virus, smallpox, that makes it quite scary to think about if you have methodologies published on how to synthesize such viruses. Does that lower the technological threshold required to synthesize related viruses? And so it's very difficult in this space to think about what other new things are going to come up in terms of biotechnology and this kind of reduced need for technical barriers that were in the past very difficult to surpass, so that you actually have, in general, more scientists, more people being able to act in this space. And going forward, I do worry that, as I showed on the first slide there of Janet Parker, that we hopefully won't, but we might be increasing our risk of having smallpox cases or something even nastier in our future. This is a quote from Professor George Church about pathogen synthesis that and as well as other genomic technologies that we can use. We might be able to take naturally occurring pathogens places that they wouldn't go on their own before. And this might be a bit sensationalist, but this is a common feeling among a lot of people within the biosecurity community. This is out of the U.S. This quote, but he's alluding to the fact that we now have capabilities that we didn't before to be able to modify pathogens to be able to make them much deadlier than what nature could. And this idea that going forward, this might be a national security concern. So it's interesting. Biological programs, looking backwards in the past, luckily have not shown as much activity or as much interest as other things such as more conventional warfare. However, that doesn't mean that there's not examples of biological warfare. So this is a biopreparate facility photographed after nearing the collapse of the Soviet Union. The Soviet program had more than 10,000 scientists and more than a dozen labs where they were synthesizing biological weapons, everything from smallpox to anthrax to tularemia. So after the collapse, they found more than 20 tons of smallpox and plague stored up that they had tried to weaponize for aerosols. Luckily, none of these, as far as we know, were ever released. However, they were very prone to accidents. So, for example, in Sverdlovsk, or sorry, I'm not pronouncing that right, Sverdlovsk, there was anthrax outbreaks in the 70s where a problem with the air purification system allowed anthrax to escape out and of their facility and kill more than 100 people in the nearby town. Thinking as well about examples, luckily, of deliberate release, there hasn't been that many examples of people with the intent to go out and use biological weapons on a mass scale. However, there are a few, and one of them is the Aum Shinrikyo cult. They, back in the 1980s and 1990s, they weaponized biological agents, unsuccessfully tried to weaponize anthrax, did successfully weaponize chemical agents such as sarin gas and kill more than 20 people and injure more than 1,000 on Tokyo subways. They had in their doctrine a mandate to want to bring about the destruction of the world. And that type of thinking combined with the capability to act upon it is something that very much causes me to pause. More recently, we know that ISIS was quite interested in biological weapons. There's been some laptops recovered from Tunisian fighters in Syria that show that they were using people that had biology and chemistry degrees to try to weaponize plague from animals that were naturally infected with plague. And they were trying to find ways that you could actually release it in air vents as well. Luckily, we don't have any recorded cases of this type of release occurring. However, it does signify that some groups are quite concerned with this as a potential weapon. It's important to note that if you work in this space, there's an issue that you can come up with, that you can be faced with regards to the fact that talking about risky things such as biological weapons can actually increase the risk, not decrease the risk. And an information hazard, as defined by Nick Brostrom, is a risk that arises from disseminating true information that may cause harm or enable an agent to cause harm. So in this sphere, it's important to remember that talking about such things is actually sometimes not a way to actually go about stopping something from happening, but instead can actually cause it to occur. An example of this is in 1925, the Geneva Protocol was written and had many nations contributed signatories to. And the 1925 Geneva Protocol banned biological and chemical weapons from being used. This is following World War I. From this, it appears in the historical record that Japan got quite interested in the idea of going, oh, if people are banning this, it must be because they're biological weapons, or it must be really good as weapons. And they seem to have been spurred by this to develop their own weapons program. And in World War II, Japan dropped plague-infested fleas, as well as used anthrax and a few other agents, and killed upwards of 400 to 500,000 Chinese, both civilians as well as military, using these biological weapons. It's unclear to me that they would have gone down this route if the discussion around how dangerous biological weapons was not so promoted. They might have not thought about it in the first place. So it's a difficult space to navigate in terms of if you care about reducing risks. I would strongly encourage, for example, EAs who are interested in this area should not sit down and think of the worst ways a lot of people could die and start writing that down and start talking about it. We don't want to do the hard work for people and give anyone any bad ideas. With regards to that as well, even if you decide, either as a scientist or as someone working in this space, that something is quite risky. So you have a bit of information that is very much something that you worry might actually increase the risk, either through, let's say, you're doing research on a virus and you're thinking about publishing the way that you can modify it to be more lethal. If you have 100 scientists who are all thinking about this, and 99 of them agree, no, I'm just not going to do this research. This is just too risky to do or to publish. All it takes is one to decide, no, I don't think it's actually that risky, and I do want to publish this, to do so and have that information released into the world. So you have this asymmetry between information release and information containment that is well summarized in this article by a colleague of mine, Greg Lewis, on the unilateralist curse, where you have unilateralist action of one who's able to supersede the beliefs of everyone else in that community. And this is something I think is quite important, especially in the EA community going forward, of having a culture of consensus when we talk about these things so that we aren't going out and increasing risks unilaterally. So just moving on to some practical tips. If this is an area that you think would be interesting to you, if you care about long-termist causes, if you think that you are particularly well suited to think about biological risks, there are things that you can do. In this field, graduate degrees are very much respected, masters and PhDs, and there's a wide range of them that you can contribute from different angles, including in the hard sciences as well as in international relations and international security. There's some areas that you can work in as well that would be quite high impact. Things like at international organizations, the WHO, the UN Office for Disarmament Affairs. National governments as well are major players in the space of health security and biosecurity. They're the major ones who are thinking about ways to mitigate some of these risks. US is a major player, but there's other governments as well too that are quite vocal on these issues. Also, think tanks as well. They can be quite influential for changing policy. Nuclear Threat Initiative is one. They have a growing biosecurity team there that they set up as focused on nuclear risks, but they have a biological risk wing of it that has formed as well. Johns Hopkins Center for Health Security is a major player in this area, and there's EA organizations that are quite interested in biological risks, such as where I work, which is at FHI, and I'm on the Research Scholars Program, which has yearly applications available for it. You don't also have to just work at any of these types of places. There's direct academia, there's direct research that you can do that could have a very large-scale impact. Everything from biological risk assessment, understanding these risks better, understanding the international community and the kind of risk landscape, as it were. Countermeasure development, so more direct technical work as well too would be quite useful. And yeah, that's the end of my talk. Thank you. Any questions?